Hi, I'm Alison Enters. I'm from Darien High School, and my project is titled The Effect of Microplastic Ingestion on the Health of Parasites, Cryptococcus in Periwinkles Litorina. Microplastics are any plastics smaller than 5 millimeters, and they threaten marine organisms because they're easy to ingest and hard to distinguish from other food sources such as planktons. Humans are also at risk of ingesting microplastics through the trophic transfer through seafood. Parasites are important to study because they shape their community structure by preventing the exponential growth of certain populations, and they are necessary to the functioning of their individual ecosystems. There's been limited research done on the interactions between parasites and microplastics. There have only been 2 studies done, and neither of them looked at the effects of microplastics on parasite health. In my study, I used Litorina, the common periwinkle snail, which is very abundant on Appledore Island at Shoals Marine Lab where I did my research. They have also been found to ingest microplastics, possibly through seaweed acting as a vector for microplastic ingestion. Litorina are infected by multiple parasites, but most abundantly by the parasite Cryptococcus which is a trematode with a 3-host life cycle. On the right, you can see a diagram of the simplified life cycle of Cryptococcus, and as you can see, Litorina, when they are the hosts, cercaria are released as that form of the life cycle of the parasite Cryptococcus, which will be important later. Just some significance of my research. Microplastics affect all marine organisms and have been found in all parts of the ocean, and humans are likely ingesting or inhaling microplastics. Parasites are necessary to the functioning of their ecosystem, which is why they're important to study. The interactions between microplastics, again, and parasites are unknown. There have been 2 studies done on these interactions, and this research has implications for other marine organisms. So my research question was, how does the ingestion of microplastics by Litorina affect the number of cercaria Cryptococcus released into the water column? And I hypothesized that if Litorina ingest microplastics, the number of cercaria released into the water column will decrease because microplastics will impact the health of Litorina and therefore the health of Cryptococcus. So in simpler terms, I thought that when the snails ingested microplastics, their health would be impaired, which would also impair the health of the parasite. So this is a simplified approach to my methods before I go more in depth. First, I collected my snails and determined whether or not they were infected with Cryptococcus. I then determined the algal consumption rates of each snail to see which ones would be most likely to ingest microplastics. I then quantified cercaria as a control before feeding half of the snails microplastics. After that, I quantified the cercaria again, and then the snails were, the soft body tissue was digested and filtered to look to see if microplastics were actually ingested. So I collected the Litorina at Loris Ledge, which is the top circle on the map. And while I was collecting the snails, I examined their foot color as a proxy for infection status, as it has been found that the deeper orange color of the foot is more likely to be correlated with an infected snail. And then the seaweed Olvalactuca was collected at the cribs, which is the lower circle on the map. So to determine whether or not the snails were infected, the Litorina were desiccated for 24 hours before each snail was isolated in its own tube. Then water samples were taken, and I looked for cercaria under the microscope, and any snails that were found to have the Cryptococcus cercaria were labeled infected and used in my experiment. And of the 44 snails I collected, 26 were found to be infected. So to characterize the background cercarial production of each snail, the Litorina were labeled and isolated again. They were desiccated for 12 hours overnight, and then the next day their containers were filled with seawater and they were provided a piece of Olvalactuca whose wet weight had been recorded prior to starting the trial. The snails were then allowed to graze for 24 hours, but after 6 hours, cercaria were quantified. The wet weight of the ova was recorded again after the grazing period of 24 hours, and the snails who ate the most became the experimental snails because we wanted to ensure that they would ingest microplastics. So to characterize the post-microplastic exposure cercarial production, the same methods were repeated. So the snails were desiccated for 12 hours, Their containers were filled with seawater, and they were provided a piece of Ovalactuca, whose wet weight had been recorded before the trial. The only difference was that the experimental snails were given a piece of Ovalactuca which had embedded microplastics, and these two pictures show what that looked like. Um, After the 24-hour period of grazing, the wet weight was recorded again, but for any of the pieces of ova that had microplastics adhered, they were rinsed off with deionized water before I weighed them. So for the digestion and filtration of the soft body tissue of my snails, I first anesthetized the snails in magnesium chloride. I measured their shell size with the electronic caliper shown up in the picture. Um, I then used a hammer to crack their shell and dissect their soft body tissue in one piece. Um, the volume of their soft body tissue was calculated using water displacement, um, which then I was able to calculate the amount of potassium hydroxide to add to the soft body tissue, which was 3 times the soft body volume. These samples were then incubated for 48 hours at 40 degrees Celsius. And then after that period, the samples were filtered using vacuum filtration and left to dry overnight. The next day, I looked for microplastics, both microplastics that I had fed them and ones that would have already been there. And if I found any that I hadn't fed them, I used the burn test to identify them as plastics. So these two graphs compare the difference in algal weight for trial 1 versus trial 2. So on the x-axis, it shows whether or not the group was the control group or the group fed microplastics, and the y-axis shows the difference in algal weight, which was calculated as the weight before the trial minus the weight after the trial. So in the graph for trial 1 on the left, you can see that the snails that were experimental and fed microplastics had a larger difference in the algal weight for trial 1, and that was a significant difference. And then in trial 2, which was interesting, the experimental and control groups did not have a significant difference between the difference in the algal weight after the trial, which is something that should be researched more because prior to the second trial, we had chosen snails that had greater consumption rates, but they ended up not eating more in the second trial. So for the average cercaria per 1 milliliter of water in trial 1 versus trial 2, the x-axis of this graph shows trial 1 and the y-axis is trial 2. So the red dots are the control group and the blue dots are the experimental group. And initially it looks like there could be a difference between this data, but the p-value ended up being 0.805, so there is no significant difference between the cercaria found in trial 1 and trial 2. So for the digestion and filtration of the soft body tissue of the snails, we did this to quantify the number of microplastics that the snails consumed, and this graph shows the snail on the x-axis and then the pieces of glitter that it consumed. And on average, the snails were only consuming 1 microplastic, with some consuming up to 4 and some consuming none. So initially, as I said, it looked like there was a difference in the cercarial output in trial 2 compared to trial 1, but this ended up not being statistically significant. This could be due to the extreme variability in cercarial output, both from snail to snail. It varies a lot, and then also the same snail, the day-to-day cercarial output fluctuates greatly. And also the snails did not ingest a lot of microplastics. On average, it was only 1 microplastic, which could be why there was no difference. As I said, the algae also gained weight more in trial 2, which was indicated by the positive difference in algal weight. So this could be due to the formation of a biofilm, possibly due to the conditions of the samples, but also it could be due to the fact that I desiccated the algae before I adhered microplastics to them, which could have caused an uptake of water. And something to consider is, does this happen with other seaweeds if I were to do this experiment with a different species? So again, my hypothesis was if Linerita littarea ingests microplastics, the number of cercaria released into the water column will decrease because microplastics will impact the health of Linerita littarea and therefore the health of Cryptocotyl lingua. but my data was really inconclusive, and in order to comment on this relationship, I would definitely need more research and more data points. Some sources of error and limitations of this study were the short time that I had to collect data, which was only 2 weeks, and okay, and I only produced two trials, which also limited the research. Also, as I said, the circadial production varies greatly, and you would need a very large sample size to account for this. And possibly, if this study were to be repeated, maybe a new proxy for parasite health would be helpful rather than circadial output because of this variability. So just some conclusions. More research must be done on this relationship between microplastics and parasites to really determine whether or not microplastics are impacting parasite health in marine organisms. I would need a longer study, a larger sample size, and possibly revised methods for testing this relationship. Here's some acknowledgements: Dr. Keogh, Dr. Fudge, Dr. Buck, Dr. Craig, and my teachers at Darien High School. And then here are my references.